When two or more loci influence the same phenotype, this is known as epistasis. Because there are multiple alleles of each contributing gene, there are multiple different genotypes which can lead to non-Mendelian inheritance. When one homozygous recessive gene blocks or alters the expression of alleles at the second gene, this is considered recessive epistasis. Martian's eye colors is either white, yellow, or green, and is determined by two different loci in a recessive epistatic pathway. Gene 1 and Gene 2 influence the color of the Martian's eyes. Gene 1 has alleles big A and little a, and Gene 2 has alleles big B and little b. If the genotype for Gene 1 includes a big A, enzyme A will be produced. This is responsible for a yellow intermediate. The phenotype for eye color starts off as a colorless precursor, but enzyme A is responsible for the yellow pigment, moving it forward down the pathway. If the phenotype gets to yellow and the genotype for Gene 2 includes a big B, enzyme B will react with the yellow pigment and produce this green end product pigment. This can be shown with a glass of milk and some food coloring. If we have enzyme A, we start with our colorless precursor, and we can move it down because we have a big A in our genotype. This leads to the color yellow being produced. Now, if we also have a big B, this enzyme reacts with the yellow intermediate to produce the green end product pigment. Now, if you do not have a big A, you are not able to even consider encoding enzyme for yellow pigment, which means you'll stay at a colorless precursor no matter what you have at Gene 2. This can be shown via the non-Mendelian inheritance ratio of 9:3:4 for big A, big B, big A, little b, and homozygous recessive for little a leads to white no matter what the B locus allele is. Now for this cross, we also have the same thing, just less alleles to work with. So in this, we have a 1:1:2 ratio where all of the homozygous recessive A alleles produce white and only white.